The following program is brought to you by Caltech.
Good evening once again. I hope everyone is enjoying their dessert at this point. I know for those of you that don't have it yet, the last of them are coming out, and I would just like to take this opportunity to introduce our keynote speaker, Professor Frances Arnold. She's the Dick and Barbara Dickinson Professor of Chemical Engineering and Biochemistry and Bioengineering here at Caltech, where she engineers biological molecules and systems by directed evolution. Professor Arnold received a bachelor's in mechanical and aerospace engineering from Princeton University in '79 and her PhD in chemical engineering from UC Berkeley in 1985. She's the co-inventor on 30 issued U.S. patents and has served as a science advisor to more than 10 companies, including Maxigen, Amiris, Codexis, Mascoma, and Gevo Inc., a company she co-founded in 2005 to develop new microbial routes to producing fuels and chemicals from renewable resources. Professor Arnold has received numerous awards, including most recently the prestigious Charles Stark Draper Prize of the National Academy of Engineering. She's a member of all three membership organizations of the National Academies, and she was elected to the American Academy of Arts and Sciences in 2011. Please join me in welcoming Professor Frances Arnold. Thank you.
What fun. I hope you're enjoying today as much as I have. I've been on airplanes most of the day. I came back from Colorado, where I was visiting the little company that was spun out of Caltech, based on a lot of Caltech technology 5 years ago, and I'm happy to report that the students who co-founded that company are still hard at work trying to turn it into a business. But I'll give you a little flavor for where that technology came from tonight. And I just want to say that I'm really honored to be able to be here and talk to you. I know that you just love science and technology, and you are really dying to hear a chemistry talk after dinner. And, you know, I could go on for 45 minutes, an hour, and you'd still be really thrilled. So I promise I won't do that, as tempted as I am, because an audience like this is always a lot of fun to talk to. But I'm going to give you a little, I hope, inspirational talk to tell you about biotechnology and where bioengineering is going. And I realize biology hasn't always been a big thrust of Caltech, so my feeling is I might be telling some of you at least some new things, because biology has been moving very, very quickly in the last few years. And you may learn some things tonight that will amaze you. I do this because I am amazed by biology. I'm an engineer. You heard I have a degree in mechanical and aerospace engineering and a PhD in chemical engineering. I've been an engineer for a long time, but I've always wanted to build beautiful, beautiful things. And to me, the most beautiful and functional things on this planet were engineered in the natural world. They were engineered by evolution. And I use that as an inspiration. I find that biology is a wonderful inspiration when it comes to building new chemical systems. You know, we chemical engineers, we like to convert one form of matter into another form of matter, hopefully something a little bit more useful or valuable than what you started with, but not always. And that's one of the problems in the biofuels industry. But nature has really been the master at doing that. And I've always been interested in sustainability. I worked at the Solar Energy Research Institute when Carter was president. I had to quit when the next president came on because I didn't think that there was going to be a very big future for that. Luckily, I became a chemical engineer and a biotechnologist. That was my big opportunity, because that's when the biotechnology revolution was just happening. You remember, 1953, we figured out the structure of DNA, right? That was only 1953. 20 years later, the first cloning experiments were being done. We were beginning to manipulate DNA. Now, I'm going to tell you some pretty amazing things about what we can do with that ability to manipulate DNA so that we can build parts of the biological world that will solve some of the problems that you've been hearing about today. And some of those biggest problems are obtaining fuels and chemicals in a renewable and sustainable fashion. So I work at the molecular level. Molecules are pretty darn cool. They will do all sorts of interesting things. And this is one of my favorite little cartoons of molecules that are carrying loads on their backs. These are not alive. These are cartoons developed by scientists. But when you think about what the molecules can do, for example, the catalysts of the biological world, these are the things that can take something like sugar, which is a great way of storing energy, and convert it into hydrocarbons, which is a great way to store energy and then release it inside of an automobile or a diesel truck. And so I want to be able to figure out how to convince these molecules to do something and to build an industry, a sustainable chemicals industry. So all this functionality, if you think about biology, all of this wonderful molecular functionality is encoded in DNA. Kind of a boring series of 4 letters unless you know how to read it. Chemically, DNA we understand. And in fact, it's gotten, here's an interesting piece of technology news. It is now possible to synthesize DNA so cheaply that all I have to do is type a sequence on my computer, email it off to Northern California or China or wherever else the synthesizers are, and within a few days I will get back in the mail a gene, a chromosome, eventually a whole genome. The physical DNA can now be synthesized to my specifications. And this technology has developed so rapidly over the last few years that I can order thousands of genes that I can then put into different organisms and see what new code, what new functions those genes encode. So that's an important aspect. In fact, you probably saw this headline last year in May where Craig Venter's group down in the San Diego area synthesized the entire 1 million base pair genome of a small and uninteresting bacterium, but the entire genome nonetheless. And so this chemically synthesized stuff was put back into a cell where the genome had been removed, and the cell just rebooted. It started dividing again. It's called Cynthia. The cell is called Cynthia, S-Y-N-T-H-I-A. It doesn't do anything particularly interesting, but it got Craig Venter's watermark in the DNA. And it was a technical proof of a demonstration of this ability we have now to synthesize to a specification an entire microbial genome. So I found that fascinating. What we have not been able to do, however, is write anything interesting. So we can write, we can build this DNA, but we don't know what codes for the solution to a human problem. Right? Evolution has created all this DNA over 4 billion years of innovation, that is trial and error, success and failure, to write all this beautiful music of life. We don't know how to do that. We're barely holding the pencil when it comes to sitting down at the computer and composing the DNA that will now take that organism and convince that organism to convert glucose into hydrocarbons. I don't know how to do that yet. But this is where we're heading, and this is what we want to be able to do. Darwin knew how this is done, and this is done by an engineering algorithm that very few of you have ever implemented in your engineering substrates. It doesn't work terribly well in non-biological systems, but actually evolution is a fantastic algorithm for design. And if you understand that and you can manipulate it, we can now breed at the molecular level new organisms to solve these human problems. So that's what I'm going to tell you very briefly about. So why is it so hard to write new DNA, right? If we go back to these molecules, I work at the level of individual proteins, catalysts that do these fascinating chemical conversions. this is really hard to design a new protein. I can go out to nature and I can grab the DNA that encodes a catalyst that exists in nature that's a result of this Darwinian discovery process. I can scrape it literally off the bottom of my shoe, and it's a beautiful catalyst, but it doesn't work for the problem. It doesn't do it well enough to make any money at it, for example. It solves a problem for the organism that needs this catalyst, and it does a great job at making new organisms, but it doesn't do a good job when the human wants to use it for something else. So what we'd really like to do is modify these catalysts. And what's hard is that they're complicated, right? We don't even understand how the sequence of DNA encodes an amino acid sequence that will fold into a three-dimensional structure, the so-called protein folding problem. When I was in graduate school, they told me that would be solved in 5 years. They lied. It wasn't solved, and it's still not solved. But then the problem that's even more complicated, more important, is how does that DNA encode what the molecule does? That's an even bigger question, the so-called protein function problem, and that's even further away from being solved. So I used to get a laugh from people because I would say, oh, in my scientific lifetime, these things aren't going to be solved. Well, my scientific lifetime keeps getting shorter and shorter from now, and I don't have to worry too much about having to eat my shoe when I say that. But it's sad. It's sad that we don't know how DNA encodes these functions. But the good news is that evolution does. And evolution, if we go out, what a lot of biologists love to do, biochemists love to go out and compare the sequences of molecules that exist in the natural world today. You can build family trees based on certain assumptions about how those evolve. And what we recognize by studying these things is they all came about by divergent evolution from some common ancestor a long time ago, and they've acquired interesting new properties over billions of years of natural selection. So that one works in one set of conditions, another does its chemical transformation on some other substrate, and they'll acquire interesting new properties just by random mutation and selection. All right, so that's what I do for a living. Humans have been doing this for a long time, right? We've been manipulating the natural world by so-called artificial evolution or artificial selection processes, and dogs are a very good example, but we've been modifying everything from corn to carrier pigeons by breeding it. We don't have any clue at the molecular level what's going on here. A child can do this, right? But it's an algorithmic process that is very effective, and it's created all sorts of varieties, for example, of dogs. Now, the natural mechanisms for doing artificial selection are limited, right? You've got mutagens to do random mutations in the genomes. You could use radiation. Sex is another great way of exploring different solutions to a problem. I know I can reach at least some fraction of you. This is a molecular optimization process, right? You are doing an optimization process on a string of letters, that's the DNA, and now the challenge becomes not of finding which two dogs to mate, but now I've got my sticky fingers on the wheel at every step in the process. I've got now different sequences of DNA that I can mate. And so I'm no longer limited the way to finding different mating types for evolution. I now am free to do pretty much what I want in the test tube. That gives me a lot of opportunity for invention. It gives me a lot of opportunity for failure, right? Because now I have to make the decisions, and in a complex optimization process, those decisions become rather hard to do. You know, just to give you, I know you have to have some numbers. I took all the chemistry out because I thought that would really kill the talk, but a couple of numbers. When you think about all the possibilities for writing a single protein, right? You've got 450 positions in the letters in the sentence, let's say, for a protein. There are 20 different letters for each position. You've got a lot of ways you can string together DNA to make a protein. And let me tell you, most of them write garbage. So here I am, I'm trying to do a molecular optimization on a fitness landscape, a space of sequences, possible sequences, that's far bigger than the universe by many orders of magnitude. Most of them don't encode anything interesting. So how on earth can this work? Well, obviously it does work, right? And it works because the fitness landscape is very smooth. What that means, for practical purposes, is that I can take a DNA sequence, really, literally from the bottom of my shoe, that encodes one of these rare functional proteins. I make mutations in that DNA just randomly, at just the right level, and I stick those back into an organism, and they do the hard part. They take that DNA and convert that into proteins. Then the hard part for me becomes identifying which of these new proteins, and here I've put a few numbers up there so you can just see the scale of the experiments, which of these proteins do anything interesting? Well, most mutations are deleterious. So in this simple optimization strategy, most of them go straight to the garbage can. But if you've asked the question in just the right way, and this is the challenge of it, you'll find mutations that improve the molecule for what you want it to do. And you just feed that back into the process and you iterate. It's evolution. It's making mutations and applying artificial selection over multiple generations. You have to be a little bit patient. And at the end of the process, you have an improved molecule, something that does something new. So, what does this let us do? Here I tweak biologists. There aren't too many biologists in the audience, but I tweak the biologists by saying, all those gorgeous molecules in the biological world, that's in the green space. Very small fraction of the possible molecules. And I'm really interested in all the possible ones because that's where the solution to the energy crisis lies, or the cure to cancer. Those things exist in that huge space, and I've got to find them. And we can do all sorts of interesting experiments to see what is physically possible. Just one example of how this is used, it's not my example, Roger Chen won the Nobel Prize using these methods to create a whole spectrum of fluorescent proteins that have literally revolutionized biology. Just by taking existing molecules from different kinds of corals or jellyfish, making mutations to those and looking at the colors that change. And he could create this whole palette of beautiful fluorescent proteins, which are used now throughout biology. I use it for biofuels, so I will, yes, get into the energy aspect of this. Once again, I'd like to use biology to solve a human problem. Evolution has not done that for me. Biology solves a biology problem. How do you survive and reproduce and make more of the next generation? So I'm trying to get these molecules to do very non-natural things, such as extract sugars from cellulosic materials. Enzymes do that. Proteins, the protein catalysts do that because various organisms would like to extract sugars from cellulose. So an enzyme has to come in there and break this down. And what we found, for example, is that you give them a little sex and they do a much better job than what you started with. What does that mean? That means we take DNA that encodes these enzymes from different organisms and we'll recombine them in an evolutionary search for better versions of that. And HJ+, just a silly name we gave one of them, is much more stable than any of its natural parents. And then you start making random mutations and selection upon that, and you can create versions of these enzymes that, for example, will work at much higher temperatures than any fungus has ever operated at. And once you can work at higher temperatures, then you get benefits such as faster kinetics, and you can increase the rate at which these enzymes will break down cellulose by almost an order of magnitude. And that makes a huge difference because the cost of these things is really what's limiting our ability to get, say, biofuels from readily available biomass that's, say, agricultural wastes. What we learn from this, and this is really interesting science, it's not just engineering, it's really interesting science because we can learn how molecules adapt in the laboratory and use that to teach us lessons about how they adapt in the natural world. You know, for example, antibiotic resistance is a really good example of how we are forcing adaptation at the molecular level in the natural world. And we find that proteins adapt really fast, just 2 or 3% of the sequence, and they can take on all sorts of new jobs. And just to make sure that I won't have to eat my shoe in the near future, I learned that many of the changes that these proteins acquire upon adaptation, we can't even explain them, much less predict them. They've happened far away from what we think are the active sites of these proteins. And in biology, I mean, the whole design problem comes down to the fact that details matter, right? 1 minute you're alive and 1 minute you're not. It's all in the details. You know, but it kind of matters a lot, and we don't understand these details when it comes to proteins. So the big vision in the biofuels industry now is, well, these new technologies such as DNA synthesis and synthetic biology and directed evolution allow us a level of control and design over the natural world, over biological systems, that allow us to have a vision of what we'd like microbes to make for us instead of ethanol, right? Ethanol is good for driving, not good for drinking, not necessarily so much for driving. Well, if you don't like ethanol as a fuel, why not make something that you could just drop right into an automobile, or you could use to make jet fuel from? So for example, we developed some processes at GEVO to make isobutanol. Isobutanol's got 4 carbons instead of 2 for ethanol. That means it doesn't it mixes perfectly well with gasoline, and it's not hygroscopic, so it doesn't get water into your distribution systems and cause everything to corrode. So you get really nice benefits from that. And what's really nice, say, for example, about something like isobutanol or some of the other molecules that you can make through this, is that you can target some very large fraction of the existing petrochemicals industry with these new synthetic alcohols, these new alcohols that you could make by reprogramming microbes to make them. So for example, isobutanol you can make into, essentially all the fuels in a very large fraction of the chemicals that make up the current petrochemicals industry. So you could actually take CO2 and energy in the form of sunlight and have a sustainable petrochemicals industry, at least in principle. A lot of engineering to make it happen in practice. So I've just tried to share with you some of the enabling technologies and some of the vision that makes it possible to think of reprogramming microbial life. They'll do it for sugar. I mean, they'll make products for you just if you give them a little bit of sugar, and a good reason to do it. And that's what reprogramming this DNA is all about. So the idea is that we can take renewable or widely available resources and create a sustainable chemicals industry. So I think I'll quit there with a reminder that it's made possible by an algorithm for design that works really well, even if you don't understand what it is you're trying to build. And that's thanks to Mr. Darwin. And I want to thank all the wonderful students that I've had over the years here at Caltech. This is such a special place. It's made it possible for an engineer to get in and do molecular biology and bioengineering, and I even get accused of practicing chemistry without a license, and that's what's really wonderful about Caltech, because it enables these explorations that would be very hard to do elsewhere. So I'll quit there, and happy to answer any questions.
I'm Bob Conley, class of 51. I worked the summer between my freshman and sophomore year in the plant physiology, plant genetics department, which was a cornfield out in Temple City. And we had the corn which had been exposed in Wichita and Bikini, and the lab assistants went out and self-served fertilized the corn. But I became aware of the fact that we could have a 50 or 100 pound stalk of corn that made maybe 2 to 5 pounds of starch. And the only difference I could find out was one was put together this way, and I could dissolve and put it in my mouth, and it would be broken down from starch. Put together this way, nothing could be done with it. And I've been waiting to see this done for 60 years to get the sugar out of the starch. And I've come to this event to hear that you're doing it.
Well, thank you.
What you said class of 60. You know, to relate this to the topic of the day, from a mathematical point of view, I'm a mathematician, the simplest way to eliminate the overconsumption of organic and synthetic organic fuels, burning hydrocarbons, would be to eliminate the consumers. And you're in a position to do that.
All right. I hope not. That's not the intention, but all sorts of unintended consequences to technology come about. No, this is not the gray goo scenario. We are not creating, not intentionally. Well, one thing to make you, if you're feeling a little nervous about this, let me tell you that my organisms, the things that we create in the laboratory, are really sick when they get out, right? They get eaten. It's a microbe eat microbe world. Sometimes it's a microbe eat us world, but it's mostly a microbe eat microbe world out there. And to compete, you have to be a very fit organism. When we do these manipulations in the laboratory, they're perfectly fine in a sterile environment, but out in the natural world, they can't even begin to compete. But you know, also to make you, hopefully make you feel better, almost all of the microbial processes that are used today do use genetically modified organisms. This is a very widespread technology.
Well, I'm not a computer scientist, and I evolve machines using the same algorithms of all sorts, and you really do get some very strange surprises.
You get some very strange and wonderful surprises, yes. Trevor.
I was going to ask the same question about raising the specter of stumbling upon something terrible, but yeah, I think you handled it.
Oh, well, I have so much fun. Last week I gave a talk to the producers, Hollywood movie producers. I do a lot of work for the Science Entertainment Exchange here in Los Angeles, outreach. And of course, they all want to know what are the apocalyptic scenarios and the contagions, you know, but I'm trying to show the good side of science. We've been modifying the natural world ever since we've been on this planet. There's no doubt about it. This is another mechanism to do that, but I would like to think that this is a mechanism by which we can actually afford to live on this planet, which is not so easy to do right now.
When you put the DNA into an organism to see what gets produced, is there a trick to selecting what kind of organism you need to use as the factory?
Yes, of course. This is not all just, you know, alchemy. There's a lot of rational design, so to speak, that goes into this, to choosing the organism that you're going to work with, to envisioning the pathway for getting from glucose to isobutanol. We know all the catalysts that have to catalyze all those reactions. And it's mostly in the fine-tuning of this and making it actually profitable, I guess it would be the way to say it, that the evolution comes in. It's very easy. I mean, you all know this because you've been out doing these things in the real world, trying to turn technology into a real solution. Often, especially in the biofuels area, that means you have to make a profit at it. You have to compete with pumping oil out of the ground. That's not easy to do, especially with our poor understanding, relatively poor understanding of these biological systems. What these technologies allow you to do is fine-tune without really knowing what you're doing at the molecular level. And that's what works really well. We can fine-tune these systems, and we can make them very, very efficient by breeding them.
So, some generally related, you read a lot in the press about corn and ethanol, and it's kind of hard to figure out what really happened on that. And so, you read the press, there's a lot of conflicting reports. Do you have a 30-second summary of what has happened there?
When the price of oil is low, corn to ethanol doesn't look very competitive. When the price of oil is high, we're all looking for it. I'm not sure what part of the controversy, if you want to worry about food versus fuel, there are a number of issues.
So it isn't really that good an idea. These things that you're talking about.
Well, no, corn to ethanol, first of all, we don't really want ethanol. We make ethanol in the biofuels industry because we know how to do it. We've been doing that for 10,000 years. humans really have liked ethanol. And it's only recently that we're starting to use it as a fuel, right? So we knew how to do it. So we just took what we knew how to do and we tried to scale it up. But it's not what you would, if you could sit back and design an organism, you wouldn't have it make ethanol. You'd have it make gasoline or isobutanol or something that would actually be useful.
Could you give about a 2-minute discussion as to how you actually go about this?
Oh, you really want to hear this at 9 o'clock at night, huh? You know, it's surprising. Your kids do this in their high school biology labs now. You can buy DNA over the internet. You can scrape it from the bottom of your shoe, and you can insert that DNA into microbes just by, basically permeabilizing their cell membranes. And if you write the right controls into the DNA, the cells will recognize that and start churning out the protein that encodes. And what we've learned over the last 25, 30 years is what are those control sequences. So this whole genetic engineering capability came from fundamental advances in biology that let us read and write the sequences that the organisms would do that. But the actual technology of it is really pretty simple. You just add DNA to microbes and you streak them out on a plate and you look for the ones that are starting to make the protein that you've got, which you can assay using analytical chemistry techniques. Gordon Moore came to my lab once. I was so proud of this technology. And he walked in, he said, "Wow, it's amazing what you can do with low tech." And I was offended. And then I just said, "Oh, wait a minute, that's really true. This is as low tech as it gets, because you can really literally do it in a high school biology lab." The hard part is how do you do it and not get garbage, right? It's with any optimization. You have to have a good strategy. Otherwise, there's lots of ways to make things that are completely useless.
I guess that's what I'm curious about. How do you develop your strategy? What mechanism or what organisms do you use? What do you feed them? How do you assay their effectiveness? So
that it's all unique to each problem that you're working on. And I'm not going to go into the details of that, but there's lots of information on the internet and in the papers that we've written on how do you, if you want to make a better laundry detergent, how would you go about doing that? If you want to make a biofuel, how would you go about doing that? And there are different strategies that you use.
Can you convert a perennial wild wheat into a tame perennial wheat which will grow quite a lot of grains?
I think that is harder to do in the laboratory because we don't work with these kinds of organisms that have long generation times. See, with bacteria, I can have a generation every day if I want. But with plants, you've got a many months long cycle. So yes, the Monsantos of the world are attempting to do that, but of course to create any new plant variety takes many years, longer than the PhD cycle. So we don't do that here. Has to fit within the PhD cycle. Otherwise you get some distressed 10th year PhD students.
Can you tweak the production of ethanol, corn, such that it produces isobutanol without destroying the dried distiller grain byproduct?
Oh, well, that's what, if you're making a business out of this. So the question is, corn has a lot of byproducts that are very useful. And so the idea is if you're going to make a fuel from the sugar component, you want to leave all the protein there for animal feed, for example. And the answer is yes, you can do that. Yeah, definitely.
So developing biofuels is an admirable and practical application for this understanding of genetics. If you had unlimited time and resources and an ultimate mandate, are there any sort of fantasy applications that you personally would like to use this for?
So sustainable fuels and chemicals is pretty fantasy for me. Right. That's a big one to bite off. Yeah. When you think of how much oil we're pumping out of the ground and how many wars we're fighting and the economic disruption that's going on. I think you all heard today energy is about the biggest problem that we can be working on. Sure, I could try to work on curing diseases. That's always But, it's not as big a problem as energy. Energy is going to touch everybody, everybody.
Curious because I was thinking about my daughter who's a bit of a pack rat. She wants to save anything she's ever done on the theory that maybe one day I'll find it useful. What do you do with the things that aren't producing us?
I throw them away heartlessly. So the question is, what do I do with the things that don't acquire the properties I want? They could be, anybody who does optimizations know that sometimes those bad things contain the information to make good things. I have so much bad stuff. I can't possibly, and I have a refrigerator. I mean, this isn't just strings on a computer. This is physical stuff that you have to store. So right now, we do simple, really simple, stupid strategies, but that's how evolution works. Kind of uphill walk, one step at a time. And I have no qualms whatsoever about throwing away 99.999% of my children.
Now if we can convince the rest of the world
to do it their way, theirs.
So you use the mutation approach now, but now you could send a string of DNA in and build something. Do you get tempted and go, this one I'm going to just tweak it a little and solve it that way?
Oh, tweak it a little with rational design? Yes, of course, yeah. So the question is, don't you get sick of all that random stuff? Don't you ever want to go in and tweak it and show that you can do it yourself? And the answer is yes. And 99% of the time that does not work, but I don't hear about that because the students never tell me those experiments. They just come to me and say, ha, 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 Francis, I did it by rational design and it worked. So yes, it does work. It does work, but it's not reliable. It's not a reliable strategy, whereas evolution is reliable. Think about it, no other engineering system has an algorithm that reliably optimizes something and works at all scales of complexity, from a single protein to a metabolic pathway to an organism to a whole ecosystem. There's one design algorithm that has generated and optimized all of those things. And so if you really want a reliable optimization strategy, that's the way to do it. And that has completely changed the way that people look at biology, because it used to be just an art. You had to be a great master chef in order to get a biological system to do something. Now, any engineer can do it. And that's what's so great about it.
I think we have time for maybe one more question. No? No takers? Well, then,
I would like to make one brief remark about Francis. At Caltech we have we have special, unusual students. They turn into special, unusual alumni. We also have we have special, unusual faculty. That's why we are great. But among those special people, some are, very few are very, very, very special. And Francis is one of them. It was mentioned earlier that she is a member of all three academies: National Academy of Engineering, National Academy of Science, and Institute of Medicine. In the history of those three academies, only 8 individuals have become a member of all three. And she's one of them. She also received the Draper Prize on the National Academy of Engineering, which was awarded a few months ago. And for engineers, lowly engineers, it is like the Nobel Prize. So she's a very, very special person, and she's also a delightful person, and it is a privilege to have her as a colleague.